One prevalent age-related neurodegenerative disorder is Alzheimer's disease. And in this lecture, I'm going to talk a little bit about how Alzheimer's disease was found and characterized, and some of the pathology behind what causes Alzheimer's disease. And so Alzheimer's is a non-reversible brain disorder. And the highest, most important risk factor for developing Alzheimer's is age, right? And so it's a time-dependent or age-dependent type of dementia. And dementia is really just any sort of memory loss disorder. And so typical of dementias, Alzheimer's disease is characterized by memory loss, a loss of cognitive function, as well as some behavioral changes. And Alzheimer's disease was first diagnosed by Alzheimer, a German physician, and the first Alzheimer's patient was this woman here, August Dieter, and Alzheimer made his diagnosis of August Dieter in the early 1900s. And what he noticed was not just a typical type of memory loss, but a complete loss of cognition, and it was these behavioral changes that were really so interesting to him. August Dieter was checked into what was then known as kind of a mental institution as a result of her dementia, and she died shortly after. And what's interesting is that they actually found samples from her brain, which were taken post-mortem. And in these brain tissue samples, they noticed kind of two things. And you can see these images here of the actual brain tissue from this first patient. They noticed these kind of clumps of protein here, which they characterized as what's now known as amyloid plaques, as well as these sort of tangles, which are now known as neurofibrillary tangles. And what's really, really remarkable is that her symptoms were later diagnosed using sort of the modern medical technology, and these brain tissue samples were actually sequenced for her DNA, and several mutations were identified and are currently under study. And so, like August Dieter, all Alzheimer's patients are characterized by the presence of these particular two particular cellular aggregates, one of which is amyloid plaques and the other of which is neurofibrillary tangles. And what an amyloid plaque is an aggregate of the protein amyloid beta. And amyloid plaques accumulate in all aged brains, and only in about 10 to 20% of cases do they actually lead to disease. These plaques are highly insoluble, and so once these aggregates of amyloid beta protein form, it's really hard to get them apart. And you can see here, the ones circled in red are the amyloid plaques. And so they tend to, you can see them here as well, staining dark brown. And these amyloid beta proteins that generate amyloid plaques are formed as a result of the cleavage, or basically the cutting, of another larger protein called amyloid precursor protein, or APP. And so the way that APP makes a beta protein, which can then lead to the formation of amyloid plaques, can be seen in the next couple slides. APP is a transmembrane protein, which means it has an extracellular domain as well as an intracellular domain, and then a transmembrane domain where it's crossing the plasma membrane. APP is found in neural tissues in the nervous system as well as non-neural tissues. It's actually expressed in a lot of different tissue types, and it has roles in synaptogenesis as well as dendrite growth, but a lot of its roles and its function are not really well defined. And so APP as a transmembrane protein needs to be made in the endoplasmic reticulum, and then it gets cleaved once it reaches the cell membrane into its kind of final form. And there are two options for this cleavage that can either result in the formation of amyloid plaques or not, right? And so depending on which enzymes or which secretases actually cleave the APP, that determines whether this generates amyloid plaques or doesn't, right? And so in the case of non-amyloid forming plaque pathway, APP is cleaved by a secretase called alpha secretase, and it generates P3 and an additional non-amyloidogenic extracellular protein. And in the case of cleavage by beta secretase, when APP is cut by this type of secretase, it generates the A beta protein as well as another extracellular protein here. And so really it's a little bit more complicated than that even. There's a series of 2 cleavage steps. A first cleavage by either alpha or beta secretase, and then a second cleavage by gamma secretase. And so in the sort of non-amyloidogenic situation, APP is first cleaved by alpha secretase, where it generates, and then subsequently cleaved by gamma secretase. And that leads to the formation of P3, which is non-amyloidogenic, and an AICD, or an amyloid intracellular domain. And in this case, P3, as I said, will not form amyloid plaques. And this is the pathway that's actually favored in non-neural tissues, or not in the brain and nervous system. However, in the brain, there's a different pathway that seems to be favored. And in this pathway, APP is cleaved first by beta secretase, and then subsequently by gamma secretase. But in the process of this second cleavage, gamma secretase generates the same AICD molecule, but it also generates a beta protein. And these A beta proteins are the ones that are amyloidogenic. And this is once again, the pathway that neural tissues or nervous system tissue favors. And you can see here that there are sort of different A betas. There's an A beta 38, 40, 42, and that just indicates the size of the protein. So A beta 38 is 38 amino acids long, A beta 40 is 40. The most amyloidogenic of these potential A betas that can form from cleavage by gamma secretase is A beta 42. A beta 40 seems to be okay. A beta 42 is the worst one of these, and has the most potential to form amyloids and aggregates. So the way that works is that A beta proteins, specifically A beta 42, first forms oligomers, or kind of several A beta 42 molecules together. And then those oligomers assemble into fibrils and become cross-linked together. And ultimately, multiple fibrils come together to form an amyloid plaque. And this is a highly ordered structure, and that's why it's so difficult to degrade and almost is always insoluble. What's interesting about A beta aggregation into the oligomers in the first place is that it doesn't really depend on any enzymes or any other kind of active processes. It seems to just be dependent on how much A beta there is. Once there's a concentration of A beta that's high enough, the chances of those 2 A beta molecules coming into contact with each other is pretty high, and then they start to form the oligomers. So the higher the concentration of A beta, the more likely it will form oligomers and ultimately aggregate into amyloid plaques. As of right now, there has not been a chaperone identified that can help fold the A beta protein, which is unfortunate because that might be able to prevent some formation of these fibrils and amyloid plaques. But right now, that is not something that we know about. And one thing I do want to stress is that amyloid plaques are not inside of the neurons themselves or inside of any tissue themselves. They're actually generated on the extracellular side. And so they don't exist inside the neurons of the brain, but rather in kind of the extracellular parts of the tissue. The other major pathological hallmark of Alzheimer's disease is the presence of neurofibrillary tangles. And neurofibrillary tangles, unlike amyloid plaques, start out as intracellular and then kind of become extracellular, and I'll explain how that happens. Now, instead of being made of A beta, the neurofibrillary tangles are made of a protein called tau. And tau's normal function is to stabilize microtubules. There are a lot of microtubules within a neuron. They help establish the axon as well as the dendrites. And so all those microtubules need to be kind of nicely stabilized and in place for neurons to function, which is why tau is so important. And on a normal microtubule, tau is phosphorylated with a couple phosphate groups, and that activates it to bind to microtubules and stabilize them. However, in some cases, tau becomes what's known as hyperphosphorylated, having way too many phosphate groups added to it, 10 to 20 different phosphate groups. And this actually has kind of a counterproductive effect on tau's normal function. When tau is hyperphosphorylated, it can't stabilize microtubules any longer. It actually leads to the destabilization of microtubules. And not only that, but the hyperphosphorylated tau starts to form its own fibers, right, which are called paired helical fibers, or PHFs. And then those paired helical fibers come together to form a neurofibrillary tangle. These paired helical fibers are also highly insoluble, just like amyloid plaques. And what ultimately happens is that they tend to destroy the microtubule network inside the neuron and replace all of the microtubules. And that leads to neurodegeneration, because a neuron without microtubules is not a neuron, and it does not have any functional abilities, and therefore it degenerates. And when it degenerates, the neurofibrillary tangles end up extracellular because there's no longer a cell to be inside of, right? So they start off intracellularly with hyperphosphorylated tau, which becomes fibrils, which then becomes the tangles. And those tangles ultimately lead to neurodegeneration. And as I said, the presence of both amyloid plaques and neurofibrillary tangles is seen in every patient with Alzheimer's disease, from August Dieter to anyone who's diagnosed with the disease today.